For most of human history, DNA was an untouchable blueprint dictating everything from appearance to disease susceptibility. It was a code we inherited, one we could not alter. But in the 21st century, a revolutionary discovery changed that forever. What began as a study of bacterial defense mechanisms turned into one of the most powerful genetic tools ever created. One that promised to cure diseases, reshape medicine, and redefine the very essence of life itself. But with great power comes great responsibility, and CRISPR's potential raised ethical questions the world had never faced before. Hi, I'm Ko, and today we're getting into the incredible story of CRISPR. We'll explore how it was discovered, how it won its creators a Nobel Prize, and how it promises to rewrite the rules of biology. But as we celebrate the scientific triumph, we must also confront the uncomfortable reality of what happens when science moves faster than ethics. Because CRISPR was not just a breakthrough, it was a Pandora's box, and soon the world would learn just how dangerous it could be. CRISPR's story didn't begin in high-tech laboratories, but in the microscopic world of bacteria, tiny organisms that have been fighting for their survival for billions of years. Long before humans learned how to wield science as a tool, bacteria had already developed complex strategies to defend themselves against relentless viral invaders known as bacteriophages. Bacteriophages, which translates bacteria eater, are simply viruses that specifically infect bacteria. In this microscopic battlefield, survival was everything. In the late 1980s, while studying Escherichia coli or E. coli, Japanese scientist Yoshino and his team stumbled upon something strange, a repeating sequences of DNA separated by unique spatial sequences. These peculiar patterns didn't seem to fit what was known about bacterial genetics. At the time, Yoshino's team wasn't sure of their significance. The discovery was briefly noted and largely overlooked by the broader scientific community. For more than a decade, these sequences remained a mystery, an oddity buried in bacterial DNA. It wasn't until the early 2000s that Spanish scientist Francisco Mojica made a groundbreaking observation that these spatial sequences matched the DNA of viruses that had previously infected the bacteria. It was as if the bacteria had developed a genetic memory, cataloging the viruses they had survived. Mojica realized he had uncovered part of the ancient bacterial immune system, an elegant defense mechanism that allowed bacteria to recognize and destroy viruses that had attacked them before. This system was later named CRISPR, short for clustered regularly interspaced short palindromic repeats, but CRISPR alone wasn't the full picture. Scientists soon discovered that bacteria also use specialized enzymes called Cas proteins, short for CRISPR-associated proteins, to cut up the viral DNA. The most famous of these enzymes, Cas9, acts like a pair of molecular scissors, precisely snipping DNA at targeted locations. At this point, the scientific world realized that what had once seemed like a biological curiosity was in fact a sophisticated genetic defense system, one that could be harnessed for purposes far beyond bacteria. For years, this discovery was simply a fascinating quirk of microbiology. But in 2012, Jennifer Doudna from the University of California at Berkeley and Emmanuel Charpentier from Umeå University in Sweden made a groundbreaking realization. CRISPR could be repurposed into a tool for precision gene editing. By programming CRISPR, scientists could cut and modify any DNA sequence, not just in bacteria, but in plants, animals, and even humans. It was like having a pair of molecular scissors that could rewrite genetic code with unprecedented accuracy. This was the moment CRISPR leaped from bacterial immunity to human technology. The implications were staggering. CRISPR offered the possibility of curing genetic diseases, making crops more resilient, and even eradicating inherited disorders. Scientists envisioned a future where conditions like sickle cell anemia and certain cancers could be permanently eliminated. In 2020, Doudna and Charpentier were awarded the Nobel Prize in Chemistry for their work, cementing CRISPR's place as one of the most important scientific breakthroughs of all time. The simplicity of CRISPR made it even more revolutionary. Unlike earlier gene editing methods, which were slow, expensive, and often inaccurate, CRISPR was cheap, fast, and incredibly precise. It democratized gene editing, allowing even small labs to experiment with genetic manipulation. Within just a few years, research using CRISPR exploded across the globe. But as with all great discoveries, CRISPR came with profound ethical dilemmas. How far should we go in editing human DNA? Should we only correct deadly diseases, or should we allow enhancements like stronger bodies, sharper minds, longer lifespans? And what about the unintended consequences? See, gene editing is not perfect, and even a small mistake could have downstream catastrophic effects on the human gene pool. In 2018, the world got its first glimpse of how dangerous CRISPR could be. A Chinese scientist had secretly pushed the boundaries of genetic engineering, conducting an experiment that many thought was still decades away. His goal was not just to study CRISPR's potential, he wanted to use it on human beings, and he did. The result? The birth of the first genetically modified babies. The revelation sent shockwaves through the scientific community. Had we already crossed the ethical line? Were we prepared for the consequences? We'll explore that story, what really happened, how the scientist was caught, and the backlash in our next episode. CRISPR gave humanity the power to rewrite the code of life, but the question still remains, should we? Thanks for watching, and I'll see you in the next video.
Double Helix, coded to reality.